Uh, Myself Hoturam, and I am here to. My project is resonance energy transfer between perovskite nanocrystal and core cell quantum dots. So here we discuss the what is the energy transfer. Radiative energy transfer when there is a two molecule A and D. D emit the wavelength which is h mu, and A absorb that wavelength and it is got excited, which is written here. It is observed that when the average distance between D and E is larger than the wavelength, uh, this energy transfer depend on the size of the sample and configuration with respect to emission and absorption, and it do not require any interaction between two molecules or atom or spectral overlap and the concentration. So here we discuss the resonance energy transfer. When two molecules, one is donor and one is acceptor, distance between them is less than 10 nanometer from one and another, then there is a resonance energy transfer occur, which is a non-radiative process as shown in this figure. This is the dotted line shows the resonance energy between two molecules. I already mentioned the energy transfer between fluorescence of donor fluorophore and absorption of acceptor fluorophore. So here we discuss the fluorescence resonance energy transfer, which is basically known as FRIET, in which we have two fluorophore. One is donor fluorophore and one is acceptor fluorophore. These are the non-radiative fashions. Uh, in this diagram, which have we shown here, that is the fluorescence of donor, which is I am showing here, and that is the absorption of acceptor, and there is the distance. That is the formula. This integration, which is given by this overlap, we can calculate this integration. ID is the intensity of donor fluorescence or fluorophore, you can say that, and epsilon A is the absorption coefficient of second fluorophore. As you can see there, if you give a light which is one wavelength and it is absorbed by the donor molecule, then it gives the fluorescence, and then if there is a overlap between two fluorophore, then a resonance energy transfer is occur, and this is the acceptor molecule, and we get the emission. Here we understand by the example. uh, this process is mainly we are studying for the living cells. Let us That is the example I have mentioned here. If we take two protein which is tagged with two different fluorophore, or I already mentioned, molecules are must be less than 10 nanometer apart, otherwise there will be no overlap, so it cannot be occur. So here is the key thing is there must be the overlap between emission of first fluorophore and excitation of second fluorophore. That is the necessary condition, otherwise this fluorescence resonance energy transfer cannot occur. So in this diagram we can see that this is our first fluorophore, and that is the excitation of first fluorophore, and that is the emission of first fluorophore, and here we can see that this is the excitation of second fluorophore. There is a nice overlap between these two, emission and excitation. So I am discussing here the example. We give the light for 40 nanometer, and we get the emission of first fluorophore at 490 nanometer. As we already discussed the overlap between lights, so I am here highlighted this. So we are taking, I am taking two fluorophore. One is yellow fluorescent protein, which is mentioned here, and other is cyan fluorescent protein. So here, for understanding, if we take this type of let us say this is YFP and this is CFP, and we give the light for 40 nanometer excitation, and we detect the fluorescence at 490 nanometer. It means there is no interaction between two protein. They are far apart, more than 10 nanometer apart. So here we are, this is sufficiently close. Then we give the light for 40 nanometer excitation. Then 490 nanometer, which is the fluorescence, I already shown here. This is the energy transfer, resonance energy transfer, and we get the fluorescence as 5 to 7 nanometer. Means we can say that there is the interaction between two proteins. That's why we get 4 to 7 nanometer fluorescence wavelength. So here I am discuss about perovskite nanocrystal, which is discovered in 1839, and first discovered by this Gustav Rose, and it is the name after the Russian mineralogist Peros, uh, Perovsky. So as I mentioned here, perovskite nanocrystal have the crystal structure type AbX3 composition, or I am, let us, one is the example I am mentioned here. A is the cesium methyl ammonium, and B is the lead, and X is the chloride, bromide, and iodide. This is the example. I red marked the PB because it is a toxic material. We already know that. That is the structure of ABX3. You can say that this is the body central type diagram, and A is at the center, and X 6X, which is octahedral shape, uh, uh, over a close outer side of the A. Perovskite nanocrystal have many properties. It has one important property, so which is broad and intense absorption, tunable band gap. So that's why it is used in solar cell, and it has narrow tunable emissions, and it gives the as the size vary, it gives the many colors of fluorescence. So we can use in LED and photo detectors. Why we are using nanocrystal, perovskite nanocrystal? So it has broad and intense uh, absorption perovskite, which can be tuned by the size of the particle and changing by the halide, or it gives the nice pattern from I am showing here in here. You can say that in x-axis we have the radius in nanometer. If you didn't decrease the radius, it gives red to blue fluorescence, which is shown the nice pattern of fluorescence. So that's why we are interested in this material. This diagram is taken from this slide. Or here I am discuss about simply just idea about core cell semiconducting quantum dots. It has it has molecular properties, and it is depend on its size. Core cell nanocrystal comprise the semiconductor of quantum dot core and cell with different semiconductor. Or here you can see that
Hoto,
example.
only 1 minute remaining, Hoto.
Yes, sir, I know that. First is
Please.
the cadmium sulfide, and core is cell is the made by the zinc sulfide, and also one example is mentioned here. Or you can see that the cadmium selenide is the core, and cadmium telluride is the uh, cell is here. This diagram is taken from, this is the references, and this is the acknowledgement, and thank you.